The stomach, as we have mentioned, has three main substances that work for the chemical digestion of the food: the renin, the pepsin, and the HCl. Please mind the renin. The spelling of renin is actually double N before the IN. Renin in the stomach, pepsin and HCl. Renin, pepsin, and HCl. Now, the function of these chemicals or the role of this chemical in digestion are very simple. Number one, the renin only acts as a substance that helps in coagulating protein that is present in milk. Coagulation of protein. Now, the protein in milk is actually a form of what we call carcinogen. But when it comes in contact with the protein in milk, it will be broken down to casein. Casein, which is the protein that is actually digestible. The protein in milk is actually casein, but it is always a form of carcinogen. So this will be broken away to add the casein, which will later be broken down to what we call amino acid. Now, the second one is pepsin. Pepsin is also an enzyme, and pepsin acts on protein. The function of pepsin is just to break down protein to polypeptide and peptones. Polypeptides, and peptones. They are just smaller, they are just smaller weight of protein. That is function of pepsin. And we have the third one, HCl. HCl in the stomach, said it's the pH of the HCl in the stomach is even close to 1, so very acidic. This HCl does a lot of work. One of the functions of the HCl is to provide a conducive environment for the two enzymes, renin and pepsin. In fact, pepsin is actually a form of pepsinogen. It is when it is in contact with HCl that is broken down to pepsin, and that it will not act as an enzyme. Now, the HCl also helps to destroy any germ that is present with the food that we are taking in. It will help to destroy all the germs or bacteria that might have been taken with the food. Aside from the HCl acting as a good buffer for the enzymes to act very well, it also helps to destroy the germs or the bacteria or the microorganisms that we might have taken alongside with the food. Now, when this food is partially digested in the stomach, we are now going to have what we call a food that is partially digested called chyme. Partially digested food that is called chyme. This chyme will move to the small intestine. Don't forget, it is still peristalsis movement. It will move to the small intestine. And the small intestine, that is where digestion of food is completed, as we believe and as we know. Digestion of food is completed. The small intestine, that's the small intestine. The digestion of food is completed in the small intestine. The small intestine has three main parts. We have the duodenum, jejunum, and we have the ileum. These are the three main parts of the small intestine: the duodenum, the jejunum, and also the ileum. And in these three parts, specific function or things are taking place here. And in the duodenum, that is the first part. The main part of the small intestine, as we mentioned, are the duodenum, jejunum, and ileum. Duodenum, jejunum, and ileum. The duodenum is the first part of the small intestine. The food will come in from the stomach as chyme, partially digested food, and this food will come in contact with what we call bile salts. Bile salts. The bile salt is actually alkaline in nature. The bile salt rather is actually there to neutralize the acidity of the chyme that is coming into the small intestine. This is because the food that is coming from the stomach is actually acidic. But the small intestine enzymes can only act on alkaline and can only act where the environment is alkaline. So the food must first of all be neutralized or made to have alkaline environments. So the bile salt will do the work of neutralization of the acid that is present on the food. Aside from that, the bile salt or the bile also help in the emulsification of fat. Emulsification. To emulsify means to break down. Emulsification of fat, to increase the surface area of fat. Now, aside from the bile and the bile acting as neutralizer of the acid present on the chyme, and also help in emulsifying the fat substance or the lipid that is coming with the food, the bile also create a conducive environment for the enzymes present in the small intestine to act on the food. Aside from that, also bile can also function as the bile can also function. Okay, I mentioned to you, can function as an emulsifier of fat or lipid, can function as neutralizer of the acid coming with the food, and finally create a conducive environment for the enzyme to act on. Now, in the duodenum, we have the gastric juice coming in contact with the food. The gastric juice we actually is rich in amylase, and also rich in trypsin, and also we have amylase, we have trypsin, and we also have in the duodenum is the first part, the middle part is the jejunum, and the last part is ileum. The jejunum receives the chyme, which is the partially digested food from the stomach. Now, at the duodenum, the bile salt released from the gallbladder to the bile duct will actually act on the chyme. First, to neutralize the acid present on the chyme. Secondly, to create a conducive environment for the enzymes that are present in the small intestine to act. Then thirdly, the bile will also help to emulsify, to break down fat. Now, the intestinal juice, which is known as the pancreatic juice, which is released from the pancreas, has three main enzymes, which are the lipase, amylase, and the third one is the trypase. Lipase, amylase, and the trypase. These three enzymes, they act or they function well in what we call alkaline environment. I mentioned again, lipase, amylase, and the trypsin. The trypsin is actually there to work on protein. Just to mention what these ones they act on, then we move away from duodenum. The lipase will act on lipid. Most time enzymes, the preface of an enzyme is just there to indicate the enzyme, while the preface rather is always there to indicate the substrate the enzyme is acting on. So we have the talking about lipid, fat and oil. Then amylase is another word for carbohydrates. It is always acting on carbohydrate substances or carbohydrate food. So to convert carbohydrates, food that you have taken in, all the starchy food you have taken in, convert them to maltose. So it's acting on carbohydrates, convert it to maltose. Then we have trypsin. We act on protein, and we convert the protein to what we call peptone or polypeptide, as the case may be. So that is what's happening at the duodenum. Now, the food at the duodenum now is no more called chyme. It is now referred to as chyle. Chyle. Now, the second part, which is the jejunum. Many scientists believe that the bile duct that releases the bile salt terminates at the jejunum, and from the jejunum it could diffuse to the duodenum and have some parts also at the ileum. Now, the jejunum has no major function in the digestive system, only that is where the bile duct is actually released. Then the last part is the ileum. The ileum has some enzymes that are present in it, and we have to be very careful about these enzymes. They are all released from the succus entericus. It's just like a juice also being released into the small intestine, the ileum in particular. The enzymes that are present here, we have erepsin. After erepsin, we have the lipase. We also have maltase. We have lactase. Aside from lactase, we also have invertase or sucrase. So these are the major enzymes that are present in the ileum, or present at the ileum, and they are present also, they are released from the succus entericus. Now, the ellipsin act on the peptide or the polypeptide and convert it to amino acid. The lipase act on the lipid or fat and oil and convert it to fatty acid. Maltase, we act on maltose. And convert it to glucose. And glucose. So that's telling us that maltose is a disaccharide. So having two molecules of monosaccharide called glucose, two molecules of glucose will give us maltose. So we have lactase also act on lactose. And we convert lactose to what we call glucose and galactose. Telling us also that lactose is actually the disaccharide. Then the last one that we have there, which is the sucrase, well, also called invertase. The sucrose, we add sucrase, we add on sucrose. That's the common sugar that we eat at home, the cane sugar, and to convert it to glucose and fructose. So these three, maltose, lactose, sucrose, they are disaccharides that can be broken down to monosaccharide as we have them on the board. So ellipsin, we act on protein and convert it to amino acid, while lipase, we act on lipid and convert it to fatty acid and glycerol. So we have this. At this point, at the ileum, digestion of food is completed. Now we have amino acid, we have fatty acid, we have glucose and other monosaccharides that the cells can make use of. With this, we say digestion or breaking down of food is completed. But is nutrition completed? No. Nutrition will be completed when the digested food is absorbed by the cell and also made use of by the cell. That's what we can say nutrition is completed. So how will it be completed? The ileum is well equipped with substances called villi. Villi, they are just finger-like projection at the inner membrane of the ileum, and they are having that projection to increase the surface area for absorption of digested food. So the digested food will be absorbed into the bloodstream via the ileum, the villi of the ileum. So the villi has large surface area, finger-like projection, has thin membrane, and also well supplied with capillaries or blood vessels to encourage the absorption of digested food. Villi itself is well supplied with blood vessels. It has large surface area due to the finger-like projection of this of the membrane of the inner membrane of the ileum. It is also having thin membrane. All of these are meant to facilitate absorption of the digested food into the bloodstream, and there the blood will take them to where they are needed. The food will not just move, the digested food will not just move from the ileum into the bloodstream. No, it will cause problem, because when we have too much of digested carbohydrates in the blood, it will result to what for diabetes. Too much of amino acid in the blood will also result to poisoning of the blood. So all of this must be regulated, and the regulation is done by what we call the liver. The liver has a portal which we call hepatic portal, and there's a vein that brings in digested food into the liver for screening. So the vein, the hepatic portal vein, will take the digested food into the liver for screening. Then the liver will regulate the amount of these food substances that will be released into the bloodstream, and from the blood it will be transported to different parts of the body where cells are found for effective usage. Now, excess glucose will be converted to glycogen by an enzyme known as insulin. insulin. So that's bringing us to something again. Insulin. Insulin will convert excess glucose to glycogen, while we also have excess amino acid could be broken down to what we call urea, and the process known as deamination. Excess amino acid could be broken down to what we call urea in a process called deamination, and sometimes excess amino acid could also be converted to useful substances that will be used for use in the cell for oxidation in order to release ATP. Now, fatty acid on its own will pass through the body and move to the heart, passing through what we call the lymphatic tissue, and there it will be deposited under different organs in the body as adipose fat or tissue in the body. And with this, we have a good understanding of what we call digestion. Now, the undigested food will move to the large intestine, where there will be absorption of water from the undigested food, and from there move to the rectum, and it will be released out of the body as feces in a process called defecation. Thank you for listening.